In this lecture, we turn our attention to the cardiac cycle. And the cardiac cycle is basically all the things happening in the heart as it goes about doing what it does. And first, we'll start off talking about the electrical activity happening in the heart that really is predominantly responsible for making that cycle happen. Then we'll talk a little bit about some of the pressures that are involved in that cardiac cycle, basically the pressures that are in the ventricles and the atria and how they open and close valves to move blood throughout the heart during the cardiac cycle. And then we'll lastly turn our attention to the regulation of what's called coronary output, basically how the cardiac cycle can be changed to either produce more or less blood for every given minute, or circulate more or less blood for every given minute. Okay, so first is the electrical activity of the heart. The heart is interesting in that it has two sets of cells. There's what we call the myocardial contractile cells. Those would be much like the muscle cells. But there's also the myocardial conducting cells. These are cells that are, even though they're kind of similar to muscle cells, they actually are more like neurons in that they will actually send depolarizations like an action potential throughout the heart. So the conducting cells make up about 1% of all of the cells in the myocardium, but they form these kind of roads or pathways. So you can see them kind of illustrated with yellow here. These are the different pathways of the conducting cells. And again, they will carry the electrical activity throughout the heart. We begin by talking about two sets of these cells that are pretty interesting. I mean, all the cells are interesting in that they have what's called automaticity, meaning they can actually generate their own action potentials. You know, oftentimes on a neuron, a standard neuron, what happens is that it has to receive lots of excitatory signals, and then if there's enough excitatory signals so that the voltage across the membrane reaches the threshold voltage at the axon hillock specifically, then it will send an action potential. That's how a normal neuron works, but in this case, these cells bring themselves to threshold. They don't rely on the stimulus of other cells per se. And that's specifically true here in these two regions, or these collections of cells called the SA node and the AV node. Now, if we were to look, the SA node can bring its collection of cells to threshold about 80 to 100 times per minute. So that means it can send out basically kind of an action potential like depolarization 80 to 100 times per minute. The cells in the AV node are a little bit slower to do so. They will send their depolarization about 40 to 60 times per minute. Okay. Now, because the cells that are here are sending depolarizations faster than the cells here, what happens is that as it sends a depolarization, that depolarization sweeps along these what are called internodal pathways, and then it will reach the AV node, and then the rhythm of the AV node actually kind of gets hijacked by the pulses that are sent by the SA node. So if you imagine, you know, this is sending out action potentials at a rate of zzz, 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 and this one's only zzz, zzz, zzz. These action potentials will go and they'll depolarize the AV node, and so that the AV node actually kind of becomes the same rhythm as the SA node. So because of that reason, because SA node is beating fast or sending depolarizations fast at a faster rate than the AV node, we call this the pacemaker of the heart. And this is actually what's going to be kind of the main generator of electrical activity in the heart, and what's really going to set the pace. Now, if we looked kind of a little bit closer at the collection of cells in here, and we were to study their membranes, they have a little bit of a different story to them than, like, let's say, an average neuron. An average neuron, maybe I'll just draw one again so we can review. An average neuron kind of looks like this. There's dendrites on it, there's a cell body, and then there's a long axon, right? And then there's axon terminals. Well, what happens is depolarizations will kind of be transmitted onto the dendrites, and if they are strong enough where they change the voltage right here from kind of like minus 70 or 90 up to minus, usually it's minus 55 is the threshold voltage. If we're studying the voltage across here and it started at minus 70 or 90, Usually it's, I think, 70 in a neuron. And it gets depolarized, 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 meaning the voltage across it is getting less and less and less and less, and it finally reaches threshold. Then it goes boom, and it sends an action potential. That voltage is completely dissipated through a process known as depolarization, and then quickly repolarized. And then that change in voltage sweeps across the axon and is kind of the signal to trigger the release of neurotransmitters at the end of the axon. That's a typical neuron, but in a conducting cell in the heart, we have a little bit of a different story. Let's say the resting membrane potential is closer, to 60 instead of 70. And these will actually have a steady influx of sodium ions into their cells, which will bring them to threshold, which is at about minus 40, so it's a little bit higher. And this happens automatically. So this is called a pre-potential. And in a neuron, it requires the stimulus of other neurons to bring the membrane to threshold. But in a heart cell, they have their own channels that allow sodium to enter, so they can bring themselves to threshold, and it's at a very rhythmic rate. The other big difference, too, is that the big depolarization that happens in a myocardial conducting cell is resulting from the influx of calcium. In a muscle cell or a neuron, that influx is due more to the influx of sodium. Okay, so calcium plays a little bit more of a special role here when we're talking about heart cells. And then similar to muscle and heart, the repolarization is the result of the potassium leaving the cell outflux. Okay, the big idea though is know what this word is, pre-potential. This is the ability for a conducting cell in the myocardium to bring itself to threshold and thereby send out a depolarization, an action potential like signal. Okay, so if we come back here to the heart, we said this is these are sending depolarizations at about 80 to 100 times. These are per minute. These are sending depolarizations at 40 to 60 times per minute. And the standard heart rate is about like 60 to 70 beats per minute. If you recall from our ECG lab, we looked at heart rate, and I think most of yours were in between 60 and 70. So that's a little bit less than 80. So you might be wondering, well, wait a minute. If the SA node is the pacemaker of the heart, why isn't our resting heart rate 80 to 100 times per minute? And that's a good question. The answer is that our heart is kind of continually being slowed down, usually. So even at rest, there's a little bit of an extra influence on this node to slow it down so that it's not its normal rate. And so because of that, it ends up being more like 60 to 70 beats per minute. Okay, let's continue on. The SA node sends out a depolarization. It sweeps down what are called the internodal pathways. There's several different kind of collections of cells that form these pathways. They form the anterior, middle, and posterior internodal pathways. You can just know these collectively as the internodal pathways. Internodal meaning between nodes. And then there's also one that will bring it over to the left atrium. That's called Bachmann's bundle. So at this point in time, we've swept depolarization waves all the way across the atria, both atria. Now remember, all the cardiomyocytes in here, the contractile cells, they have gap junctions between them. And so even though we're sending depolarizations across here and then across here, they end up passing, that electrical activity passes through and causes a contraction in all of the cells here of the atria because of their interconnectedness via the gap junctions that we talked about. The gap junctions, if you've forgotten, they're the little holes that exist between the membrane of neighboring myocardial cells. Now there is a little bit of an insulator kind of here at the atria so that those depolarizations that have occurred in the atrial cells do not get transmitted down to the ventricles. Okay, and that's actually pretty important. Okay, once the depolarization then is swept throughout the atria, then from the AV node it goes down this little bundle here, which is called the bundle of His, or the atrioventricular bundle, because it's taking it from the atria and bringing that signal to the ventricles. Then that signal is going to pass down the conducting cells that are found in the interventricular septum, and those are called the right and the left bundle branches. So you have one here, and you have one here. The left will then go and kind of penetrate the myocardium surrounding the left ventricle. The right will kind of go down and penetrate the myocardium of the right ventricle. And from there, we have little fibers that branch off, which are called the Purkinje fibers. And then they're going to really transmit that depolarization nice and even across all of the contractile cells of the myocardium. You can also note here we have, like this is the moderator band. You see that there's, I think this is the moderator band, or maybe not, but you see that this, from here we actually get some connections into the papillary muscles, so that those papillary muscles contract and put some tension on the valves. You also get those down in here. So there's some other parts to this as well. The Purkinje fibers will also have spontaneous action potentials generated, but it's usually at about 30 times per minute, so it's not quite enough to significantly send blood throughout the body as it needs to. The cells here also have gap junctions, and so when those waves of electrical activity come to the myocardium and spread throughout the Purkinje fibers, all of these cells will kind of beat or contract as one unit. Okay. Now there is a delay though. It takes about 100 milliseconds for the signal to get from the AV bundle all the way down to the bundle branches and into the Purkinje fibers. What that means is that the atria will always contract first, then there will be about a 100 millisecond delay, and then the ventricles will contract. And that's important because the job of the atria is to send blood into the ventricles and to pack the ventricles as full as they can before the ventricles contract. And if the atria and the ventricles contracted at the same time, blood would not go from point A to point B. The heart would just kind of seize and blood would just kind of not really go anywhere. But if these contract first, it pushes the blood in, packs it full, and then there's 100 milliseconds for that to happen before the ventricles are now, which are hopefully bursting at the seams, can contract and send their blood out the aorta or the pulmonary trunk. Here's just a quick summary of what's happening here. You start with the SA node. That depolarization sweeps across all of the cells of the atria, and then there's about 100 millisecond delay as it passes down through the bundle of His and the bundle branches and into the Purkinje fibers, and then all of the contracting cells in the ventricles kind of contract as one unit, and then we start the whole process again. All right, now we can observe some of these events that are happening because it involves lots of changes in voltage. And so if we have kind of a piece of equipment that can monitor voltage changes happening in the heart, we can use it to study if all of these events are occurring as they should. This is called an ECG or an EKG. EKG is the German spelling of it. ECG is the English spelling of it. It stands for electrocardiogram. And there's several different kinds. Basically, there's you can put different leads on the body. This is, let's see, a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 lead ECG. They're showing a picture of a 10 lead one. In lab, we do only a 3 lead ECG. But in either case, some of the main features that an ECG will detect are the following. There is what's called the P wave. There's what's called the QRS complex, and then there's what's called the T wave. And these all represent different things. The P wave, this little first bump that occurs, is the change in voltage that is detected when the atria contract. The QRS complex is the change in voltage detected when the ventricles contract. And then the T wave is the change in voltage that is detected when the ventricles relax. And you might ask, well, when do the atria relax? You know, the ventricles contract and the ventricles relax, but here the atria only contract. Well, the atria relax kind of somewhere in here, but it just kind of gets swallowed up by the QRS wave. So that's kind of why they call this a QRS complex versus just a QRS wave. because there's kind of some other things that are happening in this whole process here. Now, by seeing these different events happening, we can print them out actually, and we can actually, based upon how many squares or how many squares are occupied by this printout on a graph such as this one, we can determine, how much the cardiac cycle is. So that each one of these kind of grids corresponds to a certain length of time, so you can kind of monitor, you can kind of see what the heart rate is. You can also make sure that the everything looks normal in what we would call for example, the PQ segment or the ST segment, the time that exists between the waves, and that's important. And we'll see it in a minute. You can diagnose different things, but generally you should have a P wave, and then shortly thereafter, about 0.2 milliseconds, or sorry, about 20 milliseconds or so, there should be a QRS wave, or sorry, this is about 100 milliseconds, and then there should be the T wave, and they should all kind of fall in relative space from one another. If there's a big delay between the P wave and the QRS complex, or a big delay between one cardiac cycle and the next cardiac cycle, it can tell you different things about the state of the heart. Now, when the heart is not beating with the proper rhythm and features that it should, we call that kind of an arrhythmia. Arrhythmia means without rhythm. And there's a couple different arrhythmias that are found using an ECG. One is called a second-degree block. This is usually a blockage of the either the bundle of His or the bundle branches. So if there's too much pressure put on the bundle of His or the bundle branches, for whatever reason, there can be kind of a blockage of that depolarization event. Now, the way that they're diagnosing this is that if you look, you'll see, like on this cardiac cycle, you see a P wave, a QRS wave, and a T wave. That's all good. Then here you see a P wave, but then there's no QRS wave before there's another P wave. So here, this is actually missing a QRS complex in the following T wave. So here the atria contracted and nothing happened. And then here the atria contracted, then we had a normal beat, and then there's the P wave, so the atria contracted, and then we had a normal QRS complex, ventricles contracting, and then same for here, but then here again we had another P wave without any QRS wave to follow it before another P wave came. So what's happening is that the signal is generated and sweeps through the internodal pathways, but then it doesn't make its way down into the Purkinje fibers, at least every time. About half the time it does, and half the time it doesn't. We would call that a second-degree partial block or heart block. That could be diagnosed with an ECG. Atrial fibrillation can be diagnosed. So here you see a nice QRS complex, so the ventricles are contracting nicely, but the atria are just kind of quivering. There's a lot of noise and a lot of electrical activity happening in the atria, so they're not actually having a nice synchronized single contraction. The heart are kind of just fluttering, or the atria are kind of just quivering. Now, I will say that this isn't particularly a life-threatening event per se, because the ventricles will fill about 50 to 70% of their volume on their own. So this is just through passive filling, and then it's the atria that will really fill that remaining 30 or more percent into the ventricle. So just with the ventricles contracting alone, there can be a pretty substantial amount of blood circulated through the body, but the atria again really greatly increase the efficiency of the ventricles. Now here we see what's called ventricular tachycardia. Tachycardia means a high heart rate, abnormally high, and you'll see that there's a lot. If you compare how far away these ventricular contractions are as opposed to these ventricular contractions, you see that these are happening about twice as fast. I'll also point out, our heart rate will generally increase or decrease to help change how much blood is circulated throughout the body. That's a very normal and good thing. What's kind of interesting, though, is that your maximum heart rate is going to be 220 beats per minute, about. And the reason for that is that the AV node, so if we go back here, the AV node which sits here, it just simply can't fire depolarizations faster than 220. So that ends up usually being about the top maximum heart rate in a healthy young individual. Okay, back to arrhythmias. Then we have ventricular fibrillation. This would be the same thing as cardiac arrest. So this is where they will get the paddles out and they'll do the clear in the shock. That's called a defibrillator, and they call it a defibrillator because you're trying to get the heart out of this fibrillation. So here you see no QRS complex, so the ventricles are not contracting, and that is a very serious life-threatening event because, again, the ventricles are what circulate blood through the body. And the defibrillator's hope is to kind of reset the heart rate, and by giving it one solid, really big depolarization event to hopefully get everything back on track. Then there is third-degree heart block. Here you're going to see that there's really no correlation between the P wave and the QRS wave at all. Here you see a P wave right before the QRS wave. Here you see a P wave and there's no QRS. Here you see a P wave and there's actually quite a bit of a long distance between the P and the QRS. Here you see a P wave and there's even a longer distance between the P wave and the QRS. So the heart really isn't responding normally to the P wave. The ventricles are just kind of contracting whenever, not followed routinely by the P wave. All right, moving right along, we'll talk just real brief about the specific events that we call that we name about the cardiac cycle. So we already mentioned the atria contracting, the ventricles contracting, but there's a few more subtleties to this that I'll quick point out. When the atria contract, we call this atrial systole, and that's the first thing to happen. The next thing that happens then is, and just so you're keeping an eye on here, we have atrial systole happening here, and then after that begins ventricular systole, so the ventricles contract, and then for a short while we have both atrial diastole and ventricular diastole, which is the relaxation of those chambers, but atrial diastole starts right here. So just keep in mind you have the atria contracting and then the atria relaxing. You also have the atria contracting and the ventricles contracting and then relaxing. So you kind of have to consider both what the atria and the ventricles are doing. Okay, but atrial contraction is atrial systole. Then as we progress into ventricular systole, there's two phases of it. One is called isovolumetric contraction. This is when the ventricles are contracting, but they're not changing the volume in the chamber. So they're squeezing the blood, but the blood isn't leaving yet because it has to actually reach a certain pressure before it's ejected. And we'll talk more about this in a second. So this is called isovolumetric contraction because it's contracting, but there's really no change in the volume of the chambers. But the pressure is greatly building in here, and that is going to get us to something that we call Boyle's law, which relates pressure and volume in a closed container. We'll talk about that in a second. Okay, then what happens is that we have this part called ventricular ejection, and this is where the pressure that is in the ventricles is great enough to allow the push of the fluid out of the heart, and so now the heart is still contracting, but this time this time the volume is actually decreasing. We're ejecting fluid or blood out of the heart. So here we kind of have our two parts of what we would call generally ventricular systole. All right, then we begin ventricular diastole. So that's what we see here, and that means the ventricles are now relaxed. And keep in mind the atria are also relaxed. So here we have atrial diastole and ventricular diastole occurring together. Now, at first we have what's called isovolumetric relaxation because the heart is relaxed, but there isn't any fluid entering it quite yet. So isovolumetric always means the volume stays the same. And then once the pressure has decreased enough, then we begin filling the ventricles. So these valves open and blood now passively fills the ventricle. Again, the ventricles will fill about 50 to 70% full just during this process. Once they get about 50 to 70% full, and there's going to be a lot of different factors that change how full they actually do get. We'll talk more about that later. But once they have passively filled quite a bit, then we complete the cycle and we get to atrial contraction. And we're going to shove every last little bit of blood we can into the ventricles to greatly improve their efficiency. Here's how these would all kind of shake out on a ECG graph, right? We have atrial, we start off, the atria and the ventricles are relaxed, okay, up to this point. Then the P wave again represents the beginning of atrial contraction. So the atria contract, so here we're in atrial systole, but the ventricles are still relaxed. And then once we get to here, roughly, the atria are still relaxed, and they're going to be relaxed all the way to the next P wave, but the ventricles are now contracting, so it's ventricular systole. So here's the contraction, and then the T wave represents ventricular diastole when that begins. And so then from this point on, the atria and the ventricles are both relaxed, and that's going to continue into our next cycle. These aren't, I apologize, these aren't really perfectly matched up here with the events on the ECG, but pretty close. Okay, so here's a little bit of review. Describe what a prepotential is and why it happens. Name two nodes that are found in the heart. Where are they found in the heart, and what do they do, and how are they different from one another? Describe the manner by which depolarization progresses through the myocardium. You should, in your description, use words such as internodal pathways, Bachman's bundle, the bundle of His, the bundle branches, and the Purkinje fibers. And probably some discussion about gap junctions. And then we have a couple of different events in the cardiac cycle. See if you could number these and figure out in which order they occur. All right, now we're going to look more specifically at the chambers of the heart and the different things that are happening pressure-wise inside those chambers. Really, what this will help you understand is why the valves open and close, and when they open and close, and why blood enters and exits different parts of the heart when it does. And when I look at a diagram like this, I always like to have a schematic of the chambers of the heart. It just makes it so much easier. What we're seeing in this graph is we're seeing a pressure tracing of the atria and the ventricles in the left side of the heart specifically. So we could we're looking specifically here. This will be kind of a similar story in the right side of the heart, just the pressures might be a little different. So here, this whatever this color would be, I don't know, like orangish, I guess, represents the atrial pressure. Green represents the ventricular pressure, and then the red represents the pressure that's in the aorta. So that would be right here. All right, so let's start at the beginning. At the beginning, we actually have a pretty low pressure. We have a pressure right here in the atrium of about, I don't know, this is probably 10, so this is maybe only like 4. So let's go ahead and put 4 millimeters of mercury here. That's millimeters of mercury stands for millimeters of hydrogium or mercury, and it just is a measure of pressure. Okay, so we have got about 4 in here, and then we've got about, I don't know, like 2 here. So very low pressures to start. There is always a little bit more pressure in the atria than there are in the ventricle during this short time because blood first pools into here. And there is a pressure gradient here, high here and low here relatively. I mean, these are both pretty low, but 4 is greater than 2. So that's why the blood that pools in here does naturally flow into the ventricles, and that's why we get that passive filling. Okay, then what happens is that we have a short little bump in the pressures, and we get that bump because of the contraction of the atria. So the atria contract, and they're going to push that blood into the ventricle. So that's why we get this little momentary increase in pressure, because it's this is momentarily squeezing the atria. Muscle walls are momentarily squeezing and creating a little bit of a pressure build here, and that's squirting the blood into the ventricle. All right, then what happens is right about here is when the ventricles contract. Okay, so this whole part right here would be what we would call the isovolumetric contraction of the ventricles. That's when the ventricles right here are contracting, but no volume is leaving yet. But I want you to see something quick here. Right as soon as the pressure in the ventricle overcomes the pressure in the atria, the AV valve closes. So specifically, this would be the bicuspid. So when the pressure in the atrium is greater than that of the ventricle, and throughout this slide I like to represent pressure by a big red swirly blob. So a big swirly blob means a greater pressure, and a smaller swirly blob means a smaller pressure. So like right here, you can see the pressure in the atria is more than the pressure in the ventricle, so blood is going in. And here's the pressure differential. But as soon as the ventricle contract, then this becomes bigger than this. And when you have a high pressure here and a low pressure here, that's what closes these valves. Okay, so it will force these valves shut. So this is all happening while this muscle in the ventricle is squeezing. Okay, this is isovolumetric contraction because there's still the same amount of blood in there. It's just getting, the pressure is just building and building and building. So the pressure is getting bigger, bigger, and 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 bigger. Now you might be asking, why is blood not leaving out the aorta if it's building the pressure? What's keeping the blood from seeping out? The answer is that the aortic valve is actually closed because of the fact that there is this pressure known as afterload pressure. So in the aorta, there's a pressure of about 80 millimeters of mercury, and that's because of all of the blood that's kind of been forced in through all the vessels of your body. They exert some pressure to them, and that pressure is actually pushing back like this and keeping the aortic valves closed. And it's also, remember, that same pressure which is sending blood through the coronary arteries to perfuse the heart. Well, for blood to actually get out of here, it has to overcome that 80 millimeter mercury roadblock. And so you can see that here the pressure is building, the pressure is building, the pressure is building. The reason why it's building is the muscles of the heart wall are squeezing, squeezing, squeezing. Anytime you squeeze or decrease, anytime you try and decrease an area of a closed volume, it increases the pressure. Okay, so in either case, we're building pressure. Now here, this represents the aortic pressure, and you can see it's at about 80 millimeters of mercury, just how I've drawn here. As soon as the pressure in the ventricle is greater than the pressure in the aorta, so as soon as it gets to about 81 or 82, it will fling open these valves, and now the floodgates are open and the blood can eject out of the ventricle. This is going to end the isovolumetric contraction and begin the ventricular ejection, blood leaving out of the ventricles. And so at this point, you can see that the pressure then, the pressure in the ventricle starts to take a different trajectory. Here it was like a skyrocket up, but as soon as it gets past the aortic pressure, it starts to slow down and then actually start to fall. And the reason why the pressure is decreasing and even starting to fall now is because we're losing fluid out of the ventricles. Okay, so on this graph, some of the big ideas are what's causing the valves to open and close. And again, it's anytime usually a pressure of something overcomes and overtakes the pressure of another thing. So in here, the ventricular pressure overcame the atrial pressure that opened the AV valve. Here, the ventricular pressure overcame the aortic pressure that opened the semilunar valve. And then we continue on, and then as that blood is leaving out of the heart, it's decreasing the pressure in the ventricle. And so the pressure falls, and then it eventually will go, eventually it will fall below that of the aorta, and then the semilunar valves close again, because now there's more pressure in the aorta. That 80 millimeters of mercury in the aorta is greater than the remainder of the pressure in the ventricle. So this shuts again. and then we continue to fall as the ventricle is now kind of relaxing, and then as they be relaxed so far, they fall below the pressure of the atria, and now we have this situation again where there's a little bit more pressure in the atria than the ventricle. So now these will open up again. AV valves open, and now blood starts to passively flow in. Excuse me. So that's a little bit about the cardiac cycle. You can also note that if you're listening to the heart, you'll hear the lub-dub, lub-dub, lub-dub. And what you're hearing is the lub is the AV valves shutting, and then the dub is the semilunar valves shutting. And then this whole process just begins again. This is one of those things that's helpful to have a little bit of review. So I've kind of put a question here for you. Review the different events that are happening at A, B, C, or D. Again, the hints are usually here. It's if it's where a pressure is overcoming another pressure, it's usually where the valves are opening or closing. So see if you can recall here. You also know that this part is a little bit goofy. This is called the dicrotic notch, and that's because of something that we won't talk about in this class. Okay, so you can read about it if you like. All right, so this is figure out what these are, and then see if you can remember what some of these things would be. You know, in regarding atrial or ventricular systole and diastole. This is a little bit of a complicated thing, and I'll spend some practice on this. And I will say that usually how I think about it is any point where the two pressures are overcoming one another, I try and think of what's happening before and what's happening after. What's the state before and what's the state after? That sometimes helps you figure it out. So, like I'll do, for example, B for you. Here we're looking at the ventricular pressure and the aortic pressure. So that would be the pressure in the ventricle and the pressure in the aorta. And it goes from being lesser in the ventricle and more in the aorta. So I like to kind of draw this with my little red squigglies. Here's the state before. You have a higher pressure in the aorta, a lesser pressure in the ventricle, and then what happens is that the ventricle overtakes the pressure in the aorta. So in that case, then this pressure gets built so high that it's greater than that in the aorta, and then that is going to actually open up this valve and allow the blood to rush out. So I always like to draw a picture like this, just showing an atrium and a ventricle and this AV valve and the SL, the semilunar valve, and think through what's happening pressure-wise. But in either case, practice this. There will be a question like this on the exam, so it's helpful to work through it. All right, the last thing we're going to talk about is cardiac output and how it's regulated. And this kind of gets into a lot of nuance regarding how the cardiovascular regulates itself. The cardiovascular system regulates itself. Cardiac output, how much the heart is ejecting, is a very important thing to consider, because keep in mind that the body will change how much blood the heart moves depending on the needs of the body. So there's a figure here, and take a look at this. You can see at rest, the heart ejects about 5 to 6 liters of blood every minute, and that gets like quadrupled when you go on a run and you have heavy exercise. So the heart will actually then push out close to between 17 and 30 liters per minute. So that's a huge jump, and that's important because there just needs to be more blood circulating through the cardiovascular system so that oxygen can be delivered to the muscles and CO2 can be removed from the muscles. Okay, so the cardiac output is determined by a couple of things. It's determined by how fast the heart is beating and how much blood is ejected every time the heart beats, which is called the stroke volume. We'll spend a little bit of time looking at how heart rate is changed to affect cardiac output. We'll spend a little bit of time, actually a lot of bit of time, talking about how stroke volume has changed to affect cardiac output. And this is going to get pretty convoluted and pretty complex. So before it does, I just want you to find your happy home in this simple equation, because we're always going to be coming back to this equation. And so if you get kind of really overwhelmed, just start again with this equation. This equation on the surface should be pretty intuitive. It should make a lot of sense. If you consider a heart and you consider how much blood it ejects every time it beats, and then you consider how many times it does eject that blood in a given minute, that should tell you how much blood total is ejected each minute. All right, let's get a little bit more complex. There are two factors, heart rate and stroke volume. Stroke volume is determined by this thing called EDV and ESV. We'll talk about those in a minute. Heart rate is controlled by a couple different things. One is there are a variety of hormones that can be released to change how fast the heart is beating. But there's also, and probably the predominant one, is the autonomic innervation. There are actually nerves that go from the brain down to the heart to either increase or decrease the heart rate. Remember, on the autonomic nervous system, there is two kind of branches. There's the sympathetic branch. This is all kind of a throwback to A&P1. And there's a parasympathetic branch. And remember that phrases that often get associated with these is sympathetic as fight or flight responses in the body, those requiring lots of energy and high stress situations. Parasympathetic signals that are going to be transmitted throughout your body help to kind of slow everything down in your body. We usually say rest or digest. And that's kind of the same example here. Parasympathetic is going to lower the heart rate, and sympathetic is going to raise the heart rate. Okay? So on the surface, pretty simple. Now, where these come from is the medulla oblongata. So this is a picture of the brain. In the medulla oblongata, which is this little middle region right here, there is what's called the cardioacceleratory center. And there is what's also called the cardioinhibitory center. And you can imagine that if you were to stimulate the cardioacceleratory center, it would lead to an increase in heart rate. Or if you were to stimulate the cardioinhibitory center, it would lead to a decrease in heart rate. Also, and this gets a little bit more complex, if you were to inhibit the cardioacceleratory center, that would help to lower the heart rate. Or if you were to inhibit the cardioinhibitory center, that would lead to an increase in heart rate. So a couple different ways that can happen. But these centers are located in the medulla oblongata, and they're just places where nerves will leave the brain and travel down to the heart. So there's a branch of the vagus nerve. The vagus nerve is very extensive. It goes to lots of different organs in the body, but the heart is no exception. So it goes down all the way down to the heart. And then there are the cardiac sympathetic nerves that also go down to the heart. And what these will do is they will change the rate of that spontaneous depolarization up to threshold that is occurring in the AV and the SA nodes. So here's a picture again of what happens normally in those nodes. The membrane, the voltage across the membrane in those conducting cells starts at about -60. It gets brought up to -40, and then that's the threshold required for the action potential to be sent. And when the parasympathetic nerves are firing, they will release, so that's the ones that are these pink ones here, they will release right here some neurotransmitters on the heart. This specifically is acetylcholine. And acetylcholine will work to actually hyperpolarize the membrane. What it does is it actually changes the permeability of those cells to ions. So it means that we won't go into it, but it makes it more or less permeable to ions. That's what causes these changes. You don't have to know exactly the why and how it changes that, makes these changes, but I do want you to know the end effect of acetylcholine being released on these cells is that it takes the normal level of the resting membrane potential and actually brings it down to -70. So it takes longer for these cells to reach threshold. So that's actually going to lead to a lower heart rate, because if it takes longer for those cells to send a depolarization, then there's less depolarizations that can happen in a given minute. So the beats per minute are going to be less. And in essence, the parasympathetic nerves have lowered heart rate. On the flip side, sympathetic stimulation is what happens when the nerve fibers in here will release norepinephrine, and that norepinephrine will bind, and it will actually do the opposite. It will raise the repolarization event so that the resting membrane potential, rather than being at -70, is now only at -50. So these cells only have to depolarize about 10 millivolts to make the action potential happen. So it happens a lot faster, and it's going to lead to a quicker heart rate. That's a huge way cardiac output is regulated, just simply by increasing how many times the heart beats or contracts in a given minute. Now, a real quick discussion here about Otto Loewi, as long as we're talking about it. I can't get through this lecture without talking about Otto Loewi because it's such a fascinating story. Back in Otto Loewi's day, so this would be kind of the turn of the century, they were wondering, they knew that the parasympathetic and sympathetic nerves could change heart rate, or specifically they were studying the parasympathetic nerves and how it lowered heart rate. But what they didn't know is, if we go back to this picture, they didn't know if the effect of the parasympathetic nerve on these cells was strictly an electrical one, or if it was a chemical one. You know, was it like literally that the parasympathetic nerve was just a wire that was plugging into the heart and changing its heart rate, or was it releasing some sort of chemical which then was acting on the heart to change its heart rate? And so this was the thing, the mystery that Otto Loewi was wondering about. And the story is that he goes to bed and he wakes up and he has a dream about how to do an experiment to figure out the riddle of that mystery. And he scribbles all of his notes down into a notebook frantically because it's, you know, probably 2 or 3 in the morning. And when he wakes up, he grabs his notebook to go do his experiment, and he can't read any of the stuff that he's written because it was written in such a frantic panic in the middle of the night. So he's very distraught because he had this brilliant experiment in mind about how to figure out whether it was a chemical or electrical impulse that changed heart rate, and he couldn't read his own writing. So the experiment had eluded him. So then he goes to bed again, and the next night, or at least maybe it was a couple nights after, but again he has the same dream where he dreams up the experiment, and this time he wakes up and he doesn't even bother writing down on his notes. He goes straight to the lab and conducts the experiment and figures out that in fact it is a chemical substance. He called it the vagus stuff, and he went on to win a Nobel Prize in I think either medicine or physiology for that discovery. So, all that to say, it is possible for you to dream up Nobel Prize winning experiments and then do them. Sometimes interesting stuff comes to our minds while we sleep. So, very interesting story there, Otto Loewi. His experiment, just in case you're interested, what he did is he took a heart and he had it, you know, he basically stimulated the parasympathetic nerve, and then it slowed the heart rate down. And then he took all of the fluid that the heart was in, and he transferred it to a new heart. So he transferred his famous vagus stuff, all the fluid here, over to this new heart, and it had the same effect. And so even though this heart wasn't actually connected to a parasympathetic nerve, the chemicals that were released by this parasympathetic nerve, the neurotransmitter acetylcholine, which we now know it to be, was accumulated in this fluid, and then that fluid was brought to this heart, so the acetylcholine was still in there, and it still had its same effect. So interesting tidbit there, Otto Loewi. Okay, so that's a little bit about heart rate. Again, it really has to do with the parasympathetic nerves and the sympathetic nerves, how they're being stimulated in the cardioinhibitory and cardioacceleratory center. Okay, so if you didn't catch this, if the brain wants to increase or decrease the heart rate, it will stimulate the neurons that are found here, the cell bodies found in these regions of the medulla oblongata, which will then transmit those signals via the sympathetic or parasympathetic nerves down to the heart. Okay, so now there's also some other hormones that are released, and they're pretty similar to acetylcholine and epinephrine, or norepinephrine. But principally, the biggest way is through those nerves, which also release those same chemicals, but just in neurotransmitter form, not hormone form. Okay, now the stroke volume side is a little bit more complex, and that's where we'll spend the rest of our time. To figure out how much blood a heart ejects, you have to consider two things. One's called the end diastolic volume, the other one's called the end systolic volume, and these words are scary, but in essence, they're very simple. End diastolic volume, let me just write it out here. Refers to how much blood is in the ventricle at the end of diastole. So again, I put up some pictures here. The systole is when the ventricles are contracting, squishing blood out. Diastole is when the heart's relaxed and filling. So when the heart is passively filling, right before it contracts, the instant before it contracts and is totally full with as much blood as it's possibly going to be filled with after the atria contract, we would call that the end diastolic volume, because at the very end of that relaxation period, how much volume is in that ventricle. The ESV doesn't stand for English Standard Version. Sorry, it's a Bible joke. It is going to be the end systolic volume, and that is how much blood is in the ventricle after the ventricle has completed systole, or its contraction. And so, it's pretty simple. If you consider how much blood is in the ventricle before it contracts and how much blood is in the ventricle after it's contracted, if you take the difference between those, it should lead to how much blood has left the heart. And that's exactly what this equation is. You take how much blood you started with, how much blood you ended up with, and that will give you your stroke volume. So the things that will change stroke volume are what changes the end diastolic volume, how much blood is going to be in here when it's relaxed, and how much blood is left in the ventricle after it's contracted. And this is where it gets kind of complex. There are basically three categories of types of things that will change the end diastolic volume and the end systolic volume. And they're right here. And I'll go through and I'll talk through these briefly. Preload refers to how full the ventricle is. And you can see that if we fill that ventricle full, and actually it's specifically the pressure inside the ventricle, but usually the pressure inside the ventricle is related to how full it is. So if we really fill that ventricle totally full, obviously it's going to lead to a greater end diastolic volume, right? Because if we fill that ventricle super full, there's going to be more volume in it at the end of its relaxation state. But what's interesting is that increasing the preload also changes and affects the end systolic volume. And this is through an interesting relationship called the Frank-Starling relationship, which basically describes how cardiomyocytes are more efficient when they're stretched. So if you consider, a heart, and here's the chambers, if we fill these ventricles fuller, it stretches out the myocardial cells, and when they are stretched out, they actually contract stronger. And so when they contract stronger, they push more blood out, and that means that there is less blood in the ventricle after it's contracted. So changing the preload will affect not only how much blood is in there at the end of relaxation, but also how much blood is left at the end of the contraction, because of the fact that that preload will increase the strength with which those muscle fibers contract. So kind of two ways there. Contractility refers to just how forcefully the heart contracts, and this really only affects end systolic volume. It doesn't really usually affect end diastolic volume. And it would make sense, if you have a ventricle that contracts very vigorously, that there wouldn't be as much blood left, so that would be a decrease in the end systolic volume. Or if you had a heart that contracted really wimpy, then there would actually be a lot of blood left over after the contraction, so that it would be a higher end systolic volume. And then lastly, the afterload is also pretty simple. The afterload again refers to the pressure in the outlet of the ventricle, and basically the ventricle has to overcome the pressure in this vessel before any blood can leave. So if the afterload pressure is super high, let's imagine that's not 80, but it's like 90 or 100. That means that more of the energy exerted in the ventricle has to go towards overcoming this pressure, And so if it spends so much of that energy overcoming this pressure, by the time the semilunar valve opens and blood can actually leave, most of the contraction has already occurred, so there's not as much left to kick that blood out of the heart. So if you increase the afterload, you will if you increase the afterload, you will increase how much blood remains in the heart after that contraction, because of the fact so much of that energy is going towards overcoming the afterload. If you decrease the afterload, that actually is going to decrease the end systolic volume, because if you decrease this pressure here, and it's a lot less, then the heart can overcome that pressure more easily, and then it has more energy to exert, or more energy to allocate towards pushing blood out of the heart. So that would mean that there's less remaining, so a smaller end systolic volume. Okay, so those are the three factors. Those three factors, what can change them are all of these things. Okay, so this is where the diagram starts to get really scary, but hang with me here. It's pretty intuitive stuff. Preload, we said how full the ventricle is, and this is pretty intuitive, that you would be able to increase how full the ventricle is for a couple of reasons. One, if there's more time for that ventricle to fill, it will get more full. Also, if you increase how much venous return is coming back to the heart, it will also increase. So the time that's allowed, and also the rate, or how much blood flow is returning back to the atria, will change how full the ventricle becomes. Venous return can be affected by, if you're exercising and your muscles are squeezing, it's squishing that venous blood back into the heart. Filling time, again, is more determined by heart rate, so how long there is between the two cardiac cycles. Contractility is affected by autonomic innervation, so this would be the parasympathetic and sympathetic nerves that we were talking about earlier. And then hormones, so it's kind of the same stuff that affects heart rate. If adrenaline is released in the bloodstream from the adrenal gland, that eventually makes its way to the heart, and it forces those cardiomyocytes to contract more vigorously. And then afterload is predominantly affected by vasodilation and vasoconstriction. If we're talking about the heart, and here's the main blood vessel, if there's kind of a peripheral vasoconstriction that happens, that will actually increase the pressure in this vessel, or if there's a vasodilation that happens where the vessel gets bigger, that will decrease the pressure in here. And so it makes it easier or less easy for the heart to overcome the afterload, depending on vasodilation, vasoconstriction. This vasodilation and vasoconstriction is also controlled by the medulla oblongata in part, at least if we're talking about systemic wide vasoconstriction, that which occurs along the entire cardiovascular system. That would be a signal coming from the medulla oblongata. All right, so this is kind of the full story. Let's try just a little bit of review with this. I'll also say there's kind of these other reflexes that make it even more complicated, but I'll stop it there. I think this is probably enough to address in this class. Okay, so consider this. If you get too overwhelmed with all of this, again, just think about these factors. Think about heart rate, think about stroke volume, and then in essence, there's three things that affect stroke volume. Okay, and then there's just examples of different ways that these are affected. I like to always kind of keep this more simplistic flow chart in mind anytime I'm working my way through these types of questions. So, describe how each of the following will contribute to cardiac output. Here's 7 different scenarios. And again, keep these keep this flow chart in mind, and also keep a picture of a heart in mind, because sometimes that's helpful as well. I'll do the first 2 or 3 of these, and then I'll see if you can do the remainder of them. All right. So, number 1, contraction of the muscles in the legs squeeze the veins. Would that affect heart rate? No. Would it affect afterload pressure? No, afterload has to do with usually the arteries. Would it affect the contractility? No. But it would affect the preload, because squeezing of the veins increases venous return to the heart. So all of that blood coming from the head and the trunk or the head and the upper limbs, and all of that blood coming from the lower extremities and the trunk, when those muscles and those veins get squeezed, they force that blood back here into the right atrium, or it could be back into the left atrium as well. But in either case, increasing the venous return will increase how full the ventricle gets. Okay, so are you with me so far? If you increase how much venous blood goes back to the heart, it will increase the preload. And then you just have to think about, if we increase the preload, how will that affect stroke volume? Increasing the preload will do two things. It will increase the end diastolic volume, because there's just simply more blood getting pushed into the ventricle. And then it also decreases the end systolic volume, because remember that Frank Starling law, if you stretch the myocardium out more, it contracts more vigorously and it ejects more blood, so there's less remaining in the ventricle after the contraction, so the ESV is less. So then consider, if I increase the end diastolic volume and I decrease the end systolic volume, what would be the net effect on stroke volume? The answer would be you're increasing this part, you're decreasing this part, so you're going to actually have a net effect of greatly increasing the stroke volume. And then if I've increased the stroke volume, considering this equation here, I've actually then increased the cardiac output. Because let's imagine that heart rate stayed the same, but I just had an increase in the stroke volume, well, that should, given this relationship, increase the cardiac output. So we would say all that to say that the cardiac output is going to be increased. Okay? Let's try another one. Increasing the end systolic volume. Okay, there's a lot of different things that could increase the end systolic volume, but it's not telling us that. It's just telling us generally end systolic volume has increased. Well, if we hold all of the other factors equal, so we assume that heart rate remains the same, and we assume that end diastolic volume remains the same, if we were to increase the end systolic volume, what effect would that have on the stroke volume? That would actually lower it, because, just to give an example, let's say end diastolic volume was 100 and end systolic volume was 10, and then after we increased the end systolic volume, it went from 100 to like 20. This is kind of like the before and after. You can see that in this scenario, the stroke volume would be 90 mils of blood, and here it would be 80 mils of blood. So when you increase the end systolic volume, this one here, you have the net effect of decreasing the stroke volume. Okay, so the net effect is we've decreased the stroke volume. And if we decrease the stroke volume, but we assume the heart rate remains the same, that would be a decrease in cardiac output. So the cardiac output would decrease if we increase the ESV, holding all factors equivalent. Okay, one last one I'll try, and then I'll see if you can get the rest. If we decrease the secretion of acetylcholine, that is going to be something that's released by parasympathetic nerves. And then you just have to recall what parasympathetic nerves do. Acetylcholine is released by parasympathetic nerves to lower the heart rate. Okay, so in this scenario, we've lowered the heart rate, and we'll assume that all the other factors are going to remain equivalent, which in reality they don't, but just for the sake of understanding these relationships, we'll assume that they do. Then if we lowered the heart rate and the stroke volume remained the same, the net effect would be that cardiac output will decrease. Try the rest of these, see how you do. Email me with questions. This concludes our discussion of the heart, both its anatomy and its physiology.